All right, thank you very much for the kind introduction. So my name is Hauke Baumann and I'm scientific staff member at amedes. And I think the title is really catchy. It was provided by varvis. It says one assay to rule them all. And for all of you who are familiar with J.R. Tolkien's novel, you might guess what the title is about. So there's one technology that is forged secretly and is better than sequencing technologies that we're using so far, such as short-read sequencing. On behalf of amedes, I would also like to thank the organizers for the opportunity to speak today. As some of the parts of this talk are not yet validated by us, I would like to label parts of the result as work in progress. I have to declare that my travel costs, hotel accommodation and participation fee for this conference have been covered by the organizers. Other than that, I have nothing to declare. So at amedes, we offer a very wide range of services in the medical diagnostics. And parts of it are also the germline and somatic genetics. We have around 5,500 employees. And for the purpose of this talk, I would like to focus on the germline genetics. And in germline genetics, we have different sizes of analytics, as probably all of you know, such as whole genome sequencing, which covers not only the exonic, but also intergenic and intronic parts of the genome, the whole exome sequencing with only the exonic parts. And we also have panel analytics, typically for a very narrow medical condition, such as thalassemias. And for a decade or more, short-read sequencing has been dominating this field, all of those three different assay types. And usually for short-read sequencing, as you can see on the right, they're very short reads only, that cover, for the enrichment approaches, that cover only the exonic parts. However, as probably many of you have seen this, this is a depiction of the IGV browser, and it depicts raw data, raw NGS data in this case. And raw NGS data of short reads oftentimes show these kind of artifacts. So what we see here is the same genomic region of the same patient by two different methods, the Illumina short reads and the PacBio long reads in this case. And in the upper panel, you can see some colored mismatches, some mismatched bases that don't belong here. And actually, this is an artifact that is really quite common in short-read sequencing. And this goes up to the point that a single nucleotide variant cannot be distinguished from the active gene or a part in the genome that is very homologous. So what we need to do as a routine laboratory, we need to have a complementary method such as Sanger sequencing or MLPA analysis, which is time-consuming and costly. So to bring everyone on the same level of knowledge concerning pseudogenes, usually they arose by gene duplication events or retrotransposition, and oftentimes they're in quite close proximity to the active gene. And they're characterized by a very high mutational burden. Usually they're not transcribed, but parts of the pseudogenes are very homologous, such as in the PMS2 gene, where we have exons 9 and 11 to 15, which are 98% homologous. So this really poses a big problem, as short reads are not uniquely matched to the active gene, but can also match the pseudogene with only a few bases that are different. In this depiction, we can again see a depiction of the IGV browser of the same patient and the same genomic region. And in the top picture, you can see an enrichment of certain exons. And in the lower picture, you can see PacBio long reads that we created. And you may wonder, so what else are the advantages? of long reads. Why do we use them? Why should we use them? Why is there a paradigm shift now in the sequencing technologies? So PacBio long reads offer a much improved read quality over the short reads. And this is not only due to the length, but also due to the quality, which I will show in the next slide. Phasing is only possible when we have a differentiating nucleotide in one of the reads. And this is oftentimes not the case for 100 base pair long read, but it is very often the time for PacBio long reads, or generally long reads. So we can phase, and we can tell the patient that this is a compound heterozygosity. And we can also, by using long reads, we can also streamline the laboratory processes. We don't need to have MLPAs and Sanger and short reads all the time in parallel. And we can streamline the analysis by using only one tool. And if you're having the reference genome HD38, you can display all your data just as I do in this depiction, the short read and long reads for one patient at the same time. So this can save time. And ultimately, this leads to a more accurate diagnosis, as we are not missing, for example, intronic deletions and other part structural variants that are missed by short reads, by Illumina. So the reason for the technology being better than the short reads is basically defined by this process over here. So we start with a double-stranded DNA molecule, which is linked by loop adapters with a patient-specific sequence over here. And this circularized DNA molecule then binds to the bottom of a cavity, which is over here on a so-called smart cell. And down here, there's a polymerase bound at the bottom. And this polymerase reads around the circular read over and over again to create multiple passes of the same molecule. And then this is computationally combined into a so-called hi-fi read. And this hi-fi read is of much better quality than just a short read, which I'll show in a few slides. And maybe something to remember is the P1 value that denotes when only one circular DNA molecule is within one cavity. Because you can't measure two molecules within one cavity. That would be the P2 value. So long-read sequencing technologies have been around for quite some years. And for a long time, the quality of the reads was not enough for routine diagnostics. And we as a company, we read this Nature Method paper with anxiety because this really is a landmark paper to show long-read sequencing technologies are taking over slowly. And this was around the time when also PacBio and Twist released a white paper saying that they wanted to create a dark region panel with 389 genes that are with a different process, with an enrichment process, and not a whole genome, a long read whole genome. And we selected the most interesting genes for our panel and for our use, which are listed over here, and did our first test run in the year 2023 and 5 different test runs in the year 2025. And just to give you a brief overview what we did in the lab. So in the wet lab, it's a 5-day protocol with around 9 hours of hands-on time. And we bought a PacBio review recently on which we are performing the analysis right in Hanover in our lab. And automation via the Hamilton is planned. Right now it is manually. And the raw data that we generate is then transferred to the varvis cloud, as we do it for short-read sequencing. And this is an exemplary test run of how the data look like when we create them with our custom enrichment panel. And on the left-hand side, you can see the number of reads and the accuracy as indicated by the Phred score. Probably most of you are familiar with the Phred score. It's the probability of calling an erroneous base, and the higher the value, the better the reads. And most reads gather at the highest possible at Q50, while usually for short reads, it's around Q35. On the right-hand side picture, you can see the number of reads and the read length on the x-axis. And the read length in our case is around 5 kb, while usually for a short read sequencing, it's 150 base pair. So in both quality metrics, we have a great improvement. To give you some other quality metrics, we have 8.7 million bar-coded Hi-Fi reads, which is around 43 gigabytes for 48 patients. Around 97 were bar-coded, and the P1 value that I mentioned earlier was around 55. Is expected between 50% and 70%, and this varies between the rounds. The polymerase read around the circular DNA molecule around 26 times. This is again a list of all the 24 genes that we selected for us. And as you may know, in NGS sequencing technologies, the coverage is a major quality metric. And we initially set out to have a 20-fold coverage, but surpassed this value by far. And have around 100 to 300 mean coverage across our genes. And the distribution of the coverage across the genes was also quite homogeneous. So this gives us all the most important features for SNV and a reliable CNV detection. Within our validation process, we investigated 5 different test runs with over 200 samples and over 590 variants that were completely concordant with Sanger sequencing. The rest of the reference sequence was also a 100% match, and within the exon distance less than 20. And with all those quality metrics and results, we got accredited by the DAKKS, which we are really proud of, and have already shifted all of our Sanger sequencing into long reads, and have already investigated hundreds of samples with long reads since January this year. One gene I'd like to mention is the gene CYP21A2, which caused a trouble due to the gene conversion events, and this really showcases the difficulty of such a project, but we are in the process of solving this issue as well. I'd now like to show you 5 different examples of use cases. So we have on the left-hand side a depiction of long reads, of PacBio long reads. I didn't have a depiction of short reads to show you how short those short reads are, but the distance of 577 base pairs is much too great for a 150 base pair read. So we couldn't, and we don't have any variant in between. So we couldn't show the allelic constellation for these 2 variants here. And with long read sequencing, we did reanalyze the sample and could easily show that these 2 pathogenic variants were located on different haplotypes. And thus, now we can say in the medical report that is going to the patient you have compound heterozygous variants, which is a confirmation of a diagnosis, and it's really important. These are 3 examples of very rare pathogenic stop variants of PMS2, and I'm really proud of this little collection because they're really so rare. And we could confirm all of those by long reads, as you can see. And we decided to go a step even further with one of those. So actually we analyzed all of those with paraphase, but in one of those cases we detected something a little bit unexpected. And this is why I want to show you the software paraphase. And during our validation process, varvis was kind to provide us this little software tool as well. And this little tool takes all the reads from the gene and the paralog and sorts it below each other. And then it separates all of those reads by haplotypes. So what you can see afterwards in the IGV browser are different haplotypes. In purple, it's like 1 and 2 haplotype of the pseudogene, and haplotype 1 and 2 of the active gene. And it will also tell you whether there's a deletion, obviously, then you would see only 3, or a duplication event. This is only available for a couple of genes now, but for those it really works very well. And sometimes needs some manual curation as well, but it's really worth to take a look into. And with one of those examples I just showed you, we performed a paraphase analysis. Over here you can see the pseudogene in the first top lanes. You can see the exons from the gene over here. And the pseudogenes are characterized by really big deletion over here. And the pathogenic stop variant is located on one of the haplotypes of the active gene. And if you really look closely, you can see a structure that is resembling one of the pseudogenes that is located also on one haplotype of the PMS2 active gene. So as the redirection goes from right to left, in this case, it doesn't really matter, but I think it's a really educational example, and we have to keep in mind that MLPA analysis does not detect variants that are balanced. So this is just something to keep in mind. Another example is the SGRC full gene deletion. You can see over here, it's also analyzed by paraphase. This is the native view that you will get out of the varvis software when you click it and go into the IGV browser. And by additionally loading the paraphase results, you can see all 3 haplotypes over here. The 2 pseudogene haplotypes have different pattern. You can see it like colored already, and there's one haplotype of the STC gene missing, and it's a very common. It's an autosomal recessive gene and a disorder, and but it's really common, and you can detect this very easily. And this is another example of the CFTR gene, exon 2 deletion. And one of the great advantages of long reads is also that you can have the exact breakpoints that you can also report. And this is sometimes quite important, as you know, for exons that are in frame and maybe stretch out into regions where you have different splicings. So yeah, this is really nice and really important, and is showcased in this figure very well. So as a summary of this talk, we at amedes aimed to supplement short-read NGS in routine diagnostics, and we have succeeded in reducing our Sanger sequencing samples a lot already. We are very happy to phase all those genes in the panel, and we think that the combined depiction of short and long read is time-saving, and we are very much looking forward to the future CNV detection in the next release of the varvis software to also validate this. And yeah, we are very happy to that long read sequencing offers us the opportunity to expand our product portfolio and align future diagnostics towards long reads. And with this, I would like to thank all the participants from the amedes side and also from the PacBio side, and all the varvis team, especially Mr. Liesfeld, for this very fruitful collaboration. It's really always very enjoyable. And if you're looking for a job as a human geneticist or scientific member at our team, please scan the QR code or look on our website. Thank you very much for your attention.